So you can survive at one temperature, but you cannot survive at a higher temperature or a lower temperature. Okay. So how do you generate mutations? Well, most of the time mutations are generated spontaneously, and they're caused by defects in the biological processes of cell division. So replication is one step when you induce a lot of different mutations, when you're copying your DNA. So when cells are undergoing mitosis and they're replicating their DNA, they will basically make a mistake and substitute the wrong nucleotide. Once that nucleotide has been incorporated into the DNA, it becomes fixed, and now you can't eliminate it, and it remains. So these are usually replication-induced mutations. Most of the errors that occur during replication are corrected. There is a repair pathway that will detect the error and try and repair it. But occasionally the repair pathway will miss those errors, and it's when those errors are missed that they become fixed. Another time when you get generated mutations is during segregation, in mitosis or in meiosis. It's when you have segregation in meiosis that the defects are passed on from adult to progeny, from parent to progeny. If the mutations occur in mitotic cells, you have other problems. You may have problems like cancer and things of that nature, basically. Segregation defects during mitosis, segregation defects during meiosis. So when chromosomes are either not recombining properly or not segregating. And so these meiotic defects are usually additions and deletions and large changes. And that's what we're going to focus on for today's class. And then, of course, you have environmental induced changes. So rather than spontaneous changes during biological processes, you have the environment impinging on this process as well. So you can exposure to various mutagens. And they can be chemical mutagens or ionizing mutagens, radioactivity. So, and a lot of different things happen. All I want you to remember is that these mutations lead to changes in the sequence of the DNA. You don't have to remember what is depurination, what is deamination, oxidation, what's a base analog, and what's an intercalating agent. The important thing is when you have these things happening, you either insert a nucleotide, you substitute a nucleotide, you delete a nucleotide. Any of those things can happen. You break the DNA, all of these can happen. So, and what happens usually is there is something that happens that removes the purine, okay, from a base. So, removal of an adenine or a guanine. Once that happens, the repair process fixes the defect, but it doesn't know which way to fix it. So it inserts any random nucleotide. So instead of it being an A or a G, it may insert a C or a T. And so in effect, it's basically making a mutation, inserting a mutation during the process of repair. And so you basically get it. Same thing happens here and here. So you generate these mutations get fixed in the DNA, and then they have consequences. And mutations are very common, amazingly common. And it's common to the point where it becomes, if you have problems, you can freak out, basically. Let's put it that way. There are 10 to the 13 cells in the human body. Okay? So you have a huge number of cells. Each cell, on average, receives between 1,000 and 10,000 lesions every day. So you can imagine how common this is. Okay? Almost all of these lesions or mutations or damage are repaired. And repaired correctly, and if they're not repaired, the cell is killed. That's the way the process works. Apoptosis, cell dies, new cell is generated and fills in the void. The most pervasive mutation is UV light. This is the most common mutation for humans. Okay? And basically, you generate 100,000 mutations per cell per hour of exposure to UV light. So when you go to the beach, right, if you don't protect yourself with SPF, whatever, the highest number you can think of, you're basically setting yourself up for a lot of mutations on your skin cells especially. Now, luckily, UV light doesn't penetrate deeply into the body, so it's only your skin cells that are affected. But that's good enough, it can kill you, right? So, this gives you an idea. Ionizing radiations are probably the worst because they penetrate your body in very deeply. They can go right through a lot of tissue. And they're the most toxic because they break DNA into two. So UV light makes only small changes in a single nucleotide. Ionizing radiations like X-rays and gamma rays will basically make double-strand breaks of your DNA. And so your DNA literally breaks, your chromosomes are broken, and you're in deep trouble if you can't repair them. So, chromosome instability, and this is because of ionizing radiations usually, gain or a loss of an entire arm of a chromosome. It's quite frequent actually. And 40% of imbalances in individuals, they'll be missing an arm of a chromosome, and that very often will lead to lethality, embryonic lethality. So the infant or the child will die before birth. 45% of terminal segment imbalances, tiny little segments at the end of a chromosome, have broken off and have been lost. So there was a study that was done recently, about 179 different humans were sequenced. And on average, each person is found to carry approximately 300 variants of a gene, 300 different variations in genes. And 50 of these are in genes that are implicated in genetic disorders of some kind or the other. More severe, less severe, whatever, but they are in. Now, that obviously doesn't mean that individuals have these disorders because they're diploid. One copy may be mutated, the other copy may be wild type, so they're heterozygous and they look normal and they function normally. But you can see the numbers are quite high. And each individual, on average, will have deletions or insertions of sequences. And there are almost 1 million such insertions in each individual. That's different from the next individual. So one individual may have 1 million deletions or insertions in one part of the genome, another one may have them somewhere else. But they're very, very common. And variations detected are not evenly distributed across the genome, basically. There are certain regions which are very highly variable and other regions that are not highly variable. Fortunately for us, most of the variation occurs in sequences that are repetitive and not important for function. They're not within genes, but they're outside of genes. And so the regions between genes act like a buffer to reduce the amount of damage that radiation may cause you, because the damages take place somewhere else, not within the 30,000 genes that are important. And so you can survive more easily. Okay. So there have been other studies as well, and I've talked about this study where they sequenced the whole family, and they were able to show that there were vast differences. What was really, really interesting and what was surprising was that there were 17 new mutations in the next generation. The children had 70 entirely new mutations that the parents did not have. They'd been generated during the process of replication and meiosis, basically, and then passed on to the infant and to the progeny. And so that was a big surprise for a lot of individuals, basically. And as I said, some alterations in sequence will concentrate in specific geographic locations because they might be beneficial. For example, we talked about this, sickle cell anemia. Sickle cell anemia has an advantage if you are living in an area that has malaria. If you're a heterozygous individual with sickle cell anemia, you are able to resist malarial infection more than you are if you are homozygous normal. And so sickle cell anemia is actually getting fixed in certain regions of the world where malaria is very common. And so it's a form of human evolution, actually, that has been taking place over the last thousands of years. Rare changes are population specific and their frequencies vary for each geographic population. Okay, so how do we study these mutations? You can have deletions, insertions, point mutations, all sorts of other mutations we'll come to soon. How do we go about studying them? So, if you have large changes, like an arm of a chromosome missing, or an arm of a chromosome duplicated, what do we do? So these are called gross chromosomal changes. Like large deletions, large insertions, large inversions of the DNA. So if the DNA is normally in this fashion, the segment will turn around 180 degrees. Or a translocation, it moves from one chromosome to another. These are called gross chromosomal changes. And we usually study them microscopically, under the microscope. looking at them after staining. And you've done this already. I'm going to go over it quickly. But if you have small mutations, tiny insertions or deletions or whatever, or point mutations, then you have to use what's known as recombinant DNA technology. So you're going to clone the DNA, you're going to sequence the DNA, and that's how you're going to be able to study it. And so we're going to walk our way through. We first focus on these gross chromosomal changes. This is chapter 13 in the 4th edition. And we'll work our way through this, and then we'll move on to the small mutations, which I believe is chapter 7 and chapter 9. Okay. So cytogenetics is the study of genetics by visualizing chromosomes, and that's what you use when you're looking at gross changes. So I've shown you this before. Here is human chromosome 2, I believe. And here is the chimpanzee chromosome, the gorilla chromosome, and the orangutan chromosome. And what you notice is there are 2 chromosomes in orangutans, gorillas, and chimpanzees, but these 2 chromosomes have fused together to form a single chromosome in human cells. Because they probably had some kind of a defect at this point, and the 2 ends joined together, and they became a single chromosome. And so you can see how that may happen. These are gross chromosomal changes, and you study things like this by taking cells, staining them, and analyzing the banding pattern. So you're looking at barcodes, and the banding patterns are characteristic of each chromosome, and you can analyze them and detect large changes that have taken place. So here is the karyotype, right? These are a partial karyotype of a human cell. You can see these bands. Each chromosome has a very distinct banding pattern. Chromosome 1 is different from chromosome 2, is different from chromosome 3, and so forth. So you can identify each chromosome based on banding patterns, and you can basically then ask, what happens if certain bands are missing or duplicated? And you can therefore be able to detect large changes that have taken place. Or if a chromosome arm is missing, you can detect that very quickly. So, that's not going to go through this. Okay, so let's talk about chromosome loss and gain. This is the largest change that can take place. An entire chromosome is duplicated or an entire chromosome is lost. Okay? And that's elevated gain or loss of complete chromosomes. And this is very frequent. As you've already observed with regards to Down syndrome, entire chromosomes can change. You can also get entire sex chromosomes being duplicated and get triploid sex chromosomes. The 4 chromosomes where you can survive with gross duplication or deletion of a chromosome are chromosomes 13, 18, 21, and the sex chromosomes. You do get loss and gain of entire chromosomes for the other chromosomes. That leads to death, fetal death. So, gross chromosomal changes are very frequent in tumors, metastatic tumors, in cancer. They're also very common in vitro fertilized embryos. It's because of the manipulations that go on during the process of in vitro fertilization. And finally, you see large amounts of these abnormal chromosomes and chromosome instability with age. So the older you are, the more likely you are to have these defects because meiosis becomes less. It's more error prone, basically. In males have a 2% rate of aneuploidy during gamete formation. Females have a 15% rate of aneuploidy during gamete formation. And as I said, the rate of aneuploidy has for some reason increased in humans compared to other mammals. And we don't really know why that is. But obviously something has been lost physically. So, gross rearrangements during in vitro fertilization, almost 40% of embryos that are generated, entire chromosomes are duplicated or missing during in vitro fertilization. And a lot of these embryos basically have to be discarded at that point. You can't implant them into the individual. And so, entire chromosome aneuploidy in human adults occurs mostly for these 4 chromosomes that I showed you. You can also get segments of chromosomes missing. And that's where we're going now. So we're missing a certain region of a chromosome. And that's where you can study this more easily. Okay. So, types of gross chromosomal changes. There are 4 different gross chromosomal changes that we're going to focus on. Large deletions, large duplications, large inversions, and large translocations of chromosomes. Okay. So, copy number variation. This is known as copy number variation because you're varying the number of copies of a particular segment of the chromosome. So you can obviously have copy number variation of entire chromosomes, but we're going to focus on copy number variation of chromosomal segments, parts of a chromosome that you can study under the microscope. So 5% of an individual's genome has some sort of a copy number variation. So between one individual and the next, there's a huge amount of variation. Some individuals will have segments that are duplicated. Some may have deletions in that segment. And so almost 5% of your genome has some sort of variation. And this has consequences as well. So there is a gene called synuclein, and this is involved in Parkinson's disease. It increases the amount of plaque formation in your brain, aggregation of certain proteins. And multiple copies or imbalanced copies of this gene result in an increased probability of degenerating Parkinson's disease. Many, many, many cancers will have gross rearrangements and copy number variations of segments of the gene. So causes all sorts of misexpressed genes, and which then leads to misexpression of downstream genes, and finally leads to oncogenesis and cancer. Surprisingly, there was a recent study that was done where they were analyzing identical twins, and they were sequencing identical twins. And they found that there were a number of copy number differences in identical twins. Probably happened early during development, right after fertilization when the 2 embryos split. And then during the divisions, they probably had early in development, there were differences because of mistakes during replication or segregation. And those mistakes then got fixed and then got propagated in those 2 individuals. Different tissues of a single individual will show copy number differences because during mitosis and replication, these differences can happen and they get fixed in the genome as well. So these things happen very commonly and they cause consequences, medical consequences, genetic consequences. Genetic consequences only arise when these mistakes take place in your germ cells during meiosis. If they happen in your body cells, in your somatic cells, in your adult tissues, they lead to medical problems like cancer, basically, or Parkinson's, or any of these neurodegenerative diseases of sorts like Alzheimer's. So, as I said, they also occur very often during in vitro fertilization. Okay. So here's a normal chromosome. This is a hypothetical chromosome. Let's imagine it has 5 or 6 different bands. Let's call these, these dark and light stained bands. Call them A, B, C, D, E, and F. This circle here is the centromere. Okay, and I'm just drawing it as a single line. Of course, these are diploids. But we'll initially just focus on one chromosome. If you have a deletion, sometimes deletions are also called deficiencies. They're interchangeable words. Here you have a deletion of a segment where the segment E is missing. Right? So that's a deletion where you miss a segment, basically. Duplication, the segment is duplicated. Rather simple, right? The E segment, there are 2 copies of it when there should be only 1 copy. An inversion is a segment that flips 180 degrees. So in the normal copy, you have D followed by E followed by F. In the inversion, you still have the same chromosome, but you have E followed by D and then F. So that segment E and D have flipped 180 degrees. That entire segment's been cut out, turned 180 degrees, and then dropped back into the chromosome. And then translocation involves 2 different chromosomes. So imagine you have 2 different normal chromosomes, one that goes A, B, C, D, E, F, and the second chromosome that goes H, I, J, K, L. In a translocation, this little segment of this chromosome switches with this segment of this chromosome. So you now have a translocated chromosome that goes A, B, C, D, L, and HIJKEF. So the E and the F segments were from this chromosome jumped onto this chromosome, and the L segment from this chromosome jumped onto this. And that's known as a translocation. And this is a balanced translocation because you're maintaining the balance. You haven't lost any genetic material, you haven't gained any genetic material, you've just rearranged genetic material. Same thing with an inversion. In an inversion, at the time when the inversion takes place, you don't gain any genetic material, you don't lose genetic material, you just kind of invert the genetic material in that segment. Deletions and duplications, you either gain or you lose genetic material. Okay. Right? Okay. So now we're going to focus on these 4 different gross chromosomal defects at the level of meiosis primarily. If these defects occur in your germ cells, what are the consequences for your progeny? That's the question we're going to basically focus on because we are a genetics class. We worry about transmission of chromosomes to different across generations. So, deletions. Deletions, large deletions are detected cytologically by comparing the banding patterns. So, here is a chromosome, and you can see that this segment of the chromosome from this band to this band has shortened. And so you can say there has been a deletion in this segment, deleted segment. So this band to this band, the distance here is much smaller than from here. Correct? And so that's how you would detect that. Okay, you have a certain nomenclature for talking about these gross chromosomal effects. And that's for deletion, it would look something like this. 46, that's the number of chromosomes in an individual, diploid individual. XX, indicating it's a female. DEL stands for deletion. 1 or 2 or 3 or 4 or 23 indicates the chromosome in which the deletion has taken place. So in this individual, this is a female with a deletion in chromosome 1, and the deletion was in the band Q24 to Q31. So that entire segment from 24 to 31 has been deleted. So that's how we would write this. Female with a deletion of chromosome 1 on the long arm Q between the bands 24 and 31. So in human cells, the long arm is called the Q arm, the short arm is called the P arm on a chromosome. And so this is on the long arm of chromosome 1. So this indicates chromosome number, 46. Female, deletion, chromosome number, which chromosome is taking place on, and the regions that are deleted, based on microscopy. Okay, so in many instances, deletions are extremely small, and you can't detect them by microscopy. And when we have those kinds of deletions, we'll use molecular techniques that we'll talk about later on in the class. For cytological deletions, large deletions, okay, it's very likely when you have a large deletion, you're going to delete some essential gene. Because it's a large deletion, multiple genes are going to be deleted at the same time, and it's possible and very likely that you're going to delete an essential gene. And so large deletions are not, cells are not happy when you have large deletions. If you're a homologous deletion, i.e. deletion on both copies, so you're deleted for both segments, you're basically very likely going to be dead. You will not be dead if you are a heterologous deletion or a heterozygous individual for those large numbers of deletions. So, kind of just common sense right here. Very large deletions, maybe you deleted 10 genes or 20 genes. Out of those 20, maybe 1 or 2 are essential genes. You need them for replication or repair or something. And when you delete both copies of them, well, the cell dies and you can't live. Okay, so what are the consequences of deletions in terms of meiosis and progeny and germ cells? So here's your normal cell, the gene, the chromosome. Imagine you have a deletion right here. Right? this segment is missing, you are heterozygous, you have one normal copy chromosome, one chromosome with a deletion. For crossing over, you need to pair. If you can't pair this gene with its homologue, you can't get a crossing over here, and so there's reduced crossing over in this region, right? Because it's missing. The segment's missing, so you're not going to see crossing over between them. And so it appears as though you have reduced crossing over taking place in these regions. In individuals heterozygous for deletion, pairing is disrupted, and therefore recombination is reduced in those regions. If you're heterozygous, so you have a wild type copy here and a mutant copy. That's your normal configuration. Let's imagine this is a gene B plus and gene B. B plus is the normal copy, B is the mutant copy. Now if you delete this segment of this chromosome right here, then the recessive allele will suddenly start to express itself. So that's another example that happens. So this segment is deleted right here. The normal copy is missing. Now you're simply a hemizygous individual for that gene. You have a single copy of that gene. And so you unmask a recessive allele. And so this is also called pseudodominance. Okay? This little d is acting like a dominant phenotype. It's not a real dominant. It's acting as a dominant simply because the actual dominant gene is not there in the cell. Okay, and deletions are kind of interesting. And here is an example I'm going to give you. Females. Remember, in flies, if you have two X chromosomes, you're female. If you have one X chromosome, you're male. It's the number of X chromosomes that determines sex in flies. And so there is a classic series of deletions in flies. So if you are 2X, you're a female. If you have a small deletion, you're happy and you're still a female. If you have a large deletion, you're a sick female because you're not happy because it's a very large deletion. It's a heterozygous individual, right? You're deleting only one segment of one chromosome of the homologous pair. If you have a very large deletion, you're dead. So, and the reason is there is an imbalance. You need two copies and you need approximate balance, and when you have very, very large deletions, a lot of different genes are imbalanced, basically, and a lot of different proteins are not in the right proportion. And at that point, the fly dies. It doesn't develop to maturity. And then when you go to a very, very, very large deletion, to the point when there is no second chromosome, you suddenly turn into a male. So transition takes place, basically. And so this shows you how these different deletions have different phenotypes, and you can only study them. You can't predict what's going to happen. You have to make the mutations, and then you have to analyze the mutations. Okay. Deletions lead to hemizygosity, i.e. one copy of a gene is present, pseudodominant, the other copy is missing. And organisms can tolerate hemizygosity for small deletions, but not for large deletions, because of imbalance in protein products. All right, so let's take a break here, and then we'll continue with the other gross chromosomal alterations.
Oh yeah. I dropped it. It's fine. You can see. I'm sorry.